So good morning, everybody. I'm from UAMS in Arkansas, and as you can tell, I have a very traditional Delta-like accent. So I hope you can follow me as we go through. I'm going to take you kicking and screaming back to biology classes at school. Okay, and I'm going to expand a little bit on what Nupa has already said and talk a little bit about genomics and genetics of multiple myeloma. So we're all made up of a code which makes us all very, very individual, and it ensures that we have different coloured hair, different body shapes, etc., etc. And that code, or the DNA, is organised into different segments. And we'll often hear about these different segments on the news. They're kind of highlighted here in the different coloured balls, and they're called genes. And each gene makes a protein, and it's those proteins which are our building blocks to help us with the different size cells and our different body functions. Now, we know that cancer gets caused because of damage to our genetic code, okay? And some cancers, we know why people get it. So, for instance, we know with skin cancer, often that might occur because the sun causes damage to the genetic code in the skin, okay? Now, normally, we would repair that damage that happens. But for cancer cells, for some reason, that damage isn't repaired and the cells go on to be abnormal. So as Dr. Raji mentioned, for multiple myeloma, the damage has been caused to the cells that hide in the bone marrow, so those are the plasma cells, okay? And as I say, for some reason, that damage hasn't been repaired and those cells are abnormal and have gone rogue. Now, at the moment, we don't really know why that damage occurs. We do know for certain things, so certainly petroleum, Agent Orange and things like that, we know that that can cause multiple myeloma, but for many, many people, we really have no idea why that damage occurs. So we've got all of this genetic code, which is in our cell. The actual DNA, the genetic code, is really long. For one cell, it's about 2 meters. So if you take the genetic code for somebody like me, you could actually put my whole genetic code around the solar system twice. That's how much genetic code we have to make us, okay? Now, clearly, it has to be well organized. And so to be well organized, this genetic code gets packed in the cell to form chromosomes. And you've probably heard about these as often advertised in on the television to work out your heritage as to where you've come from, and so on and so forth, okay? And so they're actually packed into the cell into 23 different pairs, okay? And this is just an example of when they've been unpacked and organized, okay, what they look like. You can see each one slightly different shape. This one is actually a lady because they've got 2 X's. If you're a gentleman, you have 1 X and 1 Y. Okay, but you should have 23 pairs and then either 2 X's or an X and a Y. But as I say, in myeloma, this genetic code has gone crazy, okay? And so what's happened is that you actually get gains of some of those chromosomes, so you get extra copies, or in some places you might get less copies, okay? Or you might just get a little bit that's broken off. Or indeed, what can happen is that the chromosomes can kind of cross over. So a little bit of one chromosome may find itself up on a bit of another chromosome, okay? Now, I must say at this point, it only happens in the myeloma cells, okay? The rest of the cells in the body are good, okay? And so one of the questions we often think about when we're talking about genetics is, am I going to pass this on to my children? Is it something I need to worry about along the family, okay? And the answer is no. These abnormal genes and proteins and chromosomes are just in those myeloma cells. So you can see here, there's a good example of a cell that's gone completely crazy. If we just take a look back at the last one, if you remember, there was 2 pairs, okay, and there's meant to be 23 and then an X and a Y. You can see here, these have been coloured to help us look at them, but like this patient's got 3 pairs of chromosome 1, it's got 3 pairs of chromosome 2, so it's gained an extra copy, whereas when you come down here, which I think is 23, you've just got 1 of that, okay? It's lost 1 of those. And the reason the colouring's helpful is that you can see that this guy here, it's supposed to be purple, okay, but it's got a bit of a blue chromosome stuck on it, possibly one from over here, okay, so they've moved around as well, okay. And so I guess the question is what advantage does a myeloma cell have if this is all going on? Why would the cell do that? We know it's damaged, okay, but the advantage that happens to a cell when this goes on is that myeloma cells, they need to be able to grow and they need to be able to survive, okay, and they need to be able to do that against the odds. In some ways, they're a little bit like weeds growing in the garden. You're meant to have flowers there, and these guys are in there taking over. They need to be able to adapt to their environment and to grow really well. And so one of the reasons why there's so many changes in there is that by doing this, they can switch off the normal way that a cell would be told to die off. So if we think about a normal healthy cell, when it gets injured or when something happens to it, it would naturally die, okay? But what happens is these myeloma cells, when they're injured, they somehow have learned not to die. So they've essentially overcome the weed killer and they can carry on growing, okay? They've also switched on genes and proteins that help them survive and help them to grow better. So they're basically turning, I was going to say, into mutants, into demons that can just grow in whatever environment they're in. So as Nupa mentioned, there's 2 tests that are kind of routinely done when you have a bone marrow test, okay? And they help us figure out what has gone wrong with an individual myeloma patient's plasma cells. And each patient will have a different problem, okay? So the cytogenetics test, it gets done on the bone marrow. It doesn't always work for everybody when they actually do the test, and that's because of the way they do the test. Because as Nupa says, the cells, they don't grow very well once you take them out of the bone marrow. They grow really well in the bone marrow, but as soon as you take them out and try and grow them in the lab to be able to do this test, they don't grow very well. So sometimes we can't find any abnormalities when we look at the cytogenetics test. The other thing about doing this test is that it's actually quite a technically challenging test, and you really need a good lab to be able to do that. And the reason I say that is if you look at this, they have to, when they look at the test, they have to decide what each of the chromosomes is. So they have to look at it and say, oh, that's chromosome 1, okay, or oh, that's chromosome 2, okay. They all are very slightly different. If you look really closely, they've got light and dark marks in different places and they're slightly different sizes. But you can imagine you really have to be an expert to be able to tell when you're looking at somebody's chromosomes what number they are, but not only that, which one's abnormal and which one's not. And I've given you a clue over on the other side. This is it's actually not a myeloma patient, but it's a good example just for the very simple reason you can see that this patient has lost a bit of chromosome 22 and it's got stuck up on chromosome 9, okay? But you have to be a real expert to be able to interpret these things, okay? When you get a loss of a chromosome like this and it gets stuck somewhere else, that's what we call a translocation, and we're going to come back to that. Okay, so the cytogenetics test is a good test. It doesn't work technically for everybody, but if it does work, you need an expert to be able to interpret the results. The test that actually gets done much more commonly is the FISH test, which Nupa talked about. And in this test, you can see here, the green is the outside of the cell. The blue is the nucleus, which is where the genetic code lies. And then what they've done is that they've put a special probe onto the cells that can make certain chromosomes shine out. So going back to what I was saying before, you should just have 2 copies of each chromosome. So you can see in this case, there's actually 3 copies. So that cell has gained a copy of a particular chromosome. Whereas in this other picture, this is a kind of an example of that translocation where one part of a chromosome moves to another part. So essentially, this is looking at a chromosome 4 and chromosome 14, and really these 2 genes should be hanging around and sticking next to each other. So you can see on this side, they're very close to each other. One's green, one's red, so it'll often look yellow, which you can see. But you can see here, these 2 guys, they've moved apart in the cell, and so they've been translocated or moved apart, and that has very specific meanings for a patient. Now, this test is much easier to do in the lab. We can, as long as the patient has plasma cells, we can do this test, okay? And it's actually technically a bit easier to interpret and to look at, because most of the time, we can all count to 3, okay? Sometimes it's not quite so simple as that, so I'm being a little bit mean, but it's much easier to look at and to interpret, okay? So what exactly are we looking for when we do these tests? And Nupam mentioned this a little bit already. But often when we first look at patients, we can split patients into 2 groups depending on what the abnormalities are in their myeloma cells. So some patients will called hyperdiploid, okay? And so that means patients have got too many chromosomes in their myeloma cells. And usually, if that's the case, it's usually the odd-numbered chromosomes that they have too many of, okay? And so that's one group of patients. The other group of patients are those ones where the chromosomes have moved around, okay? And this often happens particularly around one of the chromosome numbers, chromosome 14, which is where all of our immune and immunoglobulins and that M protein we were talking about is made. And so many patients will have a bit of chromosome 11 stuck to a bit of chromosome 14, or a bit of chromosome 4 stuck there, and so they form a different group. And so those are one of the key things we're looking at when we do these tests. And I'll tell you a little bit why we're looking for it in a moment. But as Nupam mentioned, these tests originally came out in about 1995, okay? And you can imagine the explosion in technologies. If you think back in 1995, would we be thinking about sending a test off to a company to find out whether we're kind of have African heritage or whether we have, European heritage? We would never have thought about that. And all of those technologies are being used in the medical and the myeloma world as well. And so we've learned an awful lot more about the actual genetics and the way that the myeloma cell is programmed over the last 20 years or so. And certainly what we've been able to show, okay, and I don't know if anybody has MGUS or smouldering myeloma in the room, do they? No? Okay. So that's a condition that happens before full-blown myeloma occurs. So some patients will have abnormal cells in their bone marrow, but they're not really having any problems. Okay. And so then as time goes by, myeloma can develop, and then unfortunately for some patients it can get really quite aggressive and become leukemia. And what we've discovered by doing all of these tests is that as time goes by, the number of abnormalities that happen in those myeloma cells increases. And so we know that often at the beginning, patients will just have these translocations or these gains, but as time goes by, as Nupam mentioned, it gets much more complicated, and patients begin to lose bits of chromosomes or gain bits of chromosomes, presumably because those myeloma cells are either trying to switch off a death signal or switch on a growth signal. Nupam also mentioned this a little bit about mutations. So a mutation, rather than a big change where you lose a whole chromosome, is just a really very small change. And when that small change happens, it changes a protein, and it can either make the protein work harder or it can make it switch off and not work at all. And so we can detect these changes with the kind of new technologies. And the reason I mention them is that many of the changes actually happen in other cancers. Okay, and that's actually a good thing for the myeloma world, because that means that we can borrow and use drugs which have been developed for other cancers, and maybe we can find out that they're useful for some myeloma patients. And a good example is that we're currently using a drug that was actually developed for melanoma, for skin cancer, for some myeloma patients. Now, it's only for a very small percentage of those myeloma patients, so about 4 myeloma patients out of 100, so it's not the answer, okay? But by understanding a little bit more about what makes a myeloma cell tick means that we can use drugs that have actually been developed for other diseases. Okay, so that's a little bit about the actual abnormalities. I just wanted to move on to say, why do we need to know about this, and why is it important that myeloma patients should know a little bit more about this? And I think there's 2 reasons. There's one, because it tells us a little bit about a patient's prognosis and how well they're going to do, okay? And then the second is it can help to direct our treatment decisions. Now, the first one, about a little bit about how well a patient's going to do, I have to say doctors are often not very keen on talking about this side of things, okay? And you can see why. When you're first diagnosed as a patient, you probably don't want the doctor saying, you're not going to do very well, okay? You want a positive doctor who's going to tell you, yes, things are going to be good, okay? One of the things we've actually found out is it is a bit of a moving target. So some of the things that we thought were not good news 10 or 15 years ago, we've actually discovered, now we've looked at more myeloma patients and we've got better technology so we can look at them better, that actually we were wrong, okay? And so we have to interpret the data not only in kind of an individual patient, but as Nupa says, we also need to look at it in the whole context of the patient. So sometimes you can have what looks like a bad genetic finding, but actually that, just because that bit's bad, the rest of the patient, so their kidneys, their M component, their beta-2M, and all the other things we look at are really good. Okay, so it needs to be all put in the mix. And I think one of the things that both doctors and patients often do is they hit a panic button just based on one thing rather than taking the whole concept together. Okay. The other thing about us learning more about it is that actually many of the things that we thought were bad news, so we used to think that a 414, when a bit of chromosome 4 was stuck to a bit of chromosome 14, was bad news. But actually, now we've got new drugs, those drugs seem to work really well. If anything, they work better for those patients. So as I say, it's a bit of a moving target, and we need to be very vigilant when we interpret the results. The other reason for knowing a little bit about our genetics of the myeloma cells is that, I guess until recently, we did used to treat all myeloma patients the same. Okay, it was very much a one-size-fits-all, and we know that's logical. If we just go around the room today, everybody will have their own story and will have very different stories, okay? And unfortunately, what we used to do as doctors is that we would just ram one treatment down for everybody, okay? We're now learning much, much more. I think a little bit, just to kind of save myself on this one, was we didn't have very many treatments, so we couldn't really individualize the treatment to a different patient. But now we have a whole selection of treatments, so we can start thinking about individualizing our treatments. Now that could be based on whether a patient prefers an intravenous drug or an oral drug, but it could also be based a little bit on the genetics. So what does a particular patient's myeloma cell look like, and which drug would be best for them? And I know Dr. Morgan's going to come back to this in a little bit more detail later. But one example is that 414 group I was talking about. That's the one where they've got the translocation with a bit of 4 and a bit of 14 in the wrong place. It's about 15% of myelomas. And we know they're a separate group because as a general rule, they tend not to get so much bone disease. But as I say, they respond really well to the proteasome inhibitors. So that's the Velcade or the bortezomib, the carfilzomib, Kyprolis, or the ixazomib, the Ninlaro. So we know now that certain groups of patients respond better. One of the exciting things that's happened in the last year for myeloma is that we've discovered this other group of myelomas, which is called the 11-14s. So that's got a bit of 14 stuck on 11 and a bit of 11 stuck on 14. And again, this is about 20% of myelomas. Okay, tends to be a little bit more common in amyloid and plasma cell leukemia. But as I say, what we've been doing is learning from other diseases. So if patients have chronic lymphocytic leukemia, which is CLL, they often get this drug called venetoclax. And what we've discovered in myeloma is that if you have this 11-14, it looks as if you may respond very well to the CLL drug. Now, those studies are still ongoing, okay, and we don't 100% know that that's the answer. But what we're now trying to do is to take patients that have the 11-14 and in a clinical trial, give them the venetoclax and see how well they do, because we think that might be another way of pulling out specific patients. Okay, just in closing my talk, I wanted to follow up a little bit on what Nupu was saying about clonal heterogeneity, okay? So we're now moving up to, I was going to say, probably postgraduate education now, okay? So I apologize. So I just kind of want to take a step back and I want to think about birds, okay? So we know that, it sounds crazy, birds have different length legs, different shaped beaks. Okay, the reason for that is because they naturally eat different kinds of foods. Yeah, so seeds, nuts, or whatever, and some will have short legs because they need, either on the ground or need to fly. Others are going to have long legs because they need to reach their food. Okay. And the reason that's happened is because they've adapted to being able to eat the food that's most appropriate for them. So they've called what we're calling adapted to their environment. And one of the questions that we'll often get is when a patient's diagnosed, okay, they have treatment, their myeloma goes away, but then it comes back. Why does it come back? Okay, and our thought process in that is probably because there was a cell that had remained after the first treatment. It was quiet, it was asleep, it wasn't doing anything, and it somehow managed to protect itself from the first treatment, and then it's managed to adapt and to change so that it can start growing slowly. And essentially, if you look at a myeloma patient's cells when they were first diagnosed and then at the first relapse and maybe even at the second relapse, those cells are always going to be very similar, but they're going to be just a little bit different, okay? And so my kind of example is thinking about sweets, okay, because my life revolves around chocolate. So, these sweets are clearly very different, okay? These ones are very much the same. If you look at myeloma cells, and we maybe think about this, the dark brown one might be the initial presentation one, then we go to the gold one, which may be when the patients relapse. They're still the same sweet, okay? They've just changed a little bit. Now, what we're trying to do at the moment, as Nupa is saying, and Dr. Morgan's going to talk about this in much more detail later, is try to characterize what these changes are, because that's going to be important in helping us to choose the right treatment for the right patient at the right time. But what I would say, and the technical term for this is clonal heterogeneity, okay? What I would say is it's not just between treatments that the cells change a little bit. So this is a PET scan, okay? The dark blobs are where the myeloma's active. And we know from following patients who've had treatment and have had PET scans that sometimes all of their black lesions, all of their lesions will disappear, okay? But sometimes many of the lesions will go, but there may just be one that's left, okay? And that suggests to us that the cells in that one area somehow have managed to adapt and have managed to survive our treatment. And what we're doing now, and I'm going to say patients that are here from Little Rock will know, is that we're trying to figure out what's different about those cells and why have they managed to survive, okay? Because that's going to be important for us as we try and plan our treatment moving forward. So, just to summarize, hopefully what I've said is that not all myelomas are the same. Okay, we know that when we chat to other patients in clinic, everybody has a unique story, but actually we know it when we look at those cells in the lab as well. Okay, and each myeloma has a different set of abnormalities, and those abnormalities can change the way that a myeloma patient responds to treatment and what their actual journey's like. And the new technologies that are coming through all the time are helping us to understand this a little bit better. And I think importantly, as Nupa mentioned, up until now, we've always had to go where the money is, which is where the myeloma cells are in the hip bone, okay? But because the technologies are getting better, we're now beginning to able to look using other things like blood tests and like scans. But certainly our kind of outstanding questions and the questions that we're hoping to answer, and I know you guys want us to answer over the next couple of years, is when you know what my myeloma is like under the microscope, which is the best treatment for me? Okay. And then the second question, which I think is probably a more difficult question, how can we stop these myeloma cells adapting and changing so that they can continue to survive in the bone marrow and become resistant to therapy, okay? And that's one that we're working quite hard on in the lab as well. So that's me, okay? And we'll take questions later, but I'm going to hand over to Dr. Morgan for his talk. Thank you.